Hello everyone, I'm Qingyu Chen from National Library of Medicine, National Institutes of Health. I'm a postdoc fellow from Dr. Zhiyong Lu's lab. Today, it's my great pleasure to talk about representing the semantics of biomedical words, concepts, and sentences regarding with their models and applications, specifically in the biomedical domain. So the aim of the talk is twofold. First, I will introduce Bioconcept work. There's a large-scale biomedical concept embeddings that we recently developed. I will also briefly cover the existing embedding models that we developed before, such as BioWordVec, that's for word embeddings, and also BioSentVec, that's for sentence embedding. You have probably noticed that I mentioned the term embedding multiple times now. So what does exactly it mean? Embedding, simply speaking, tries to capture the semantic meaning of words, concepts, and sentences in a fixed vector. For example, let's say word embedding. It tries to capture the semantic meanings of the words mentioned in the literature in a fixed size of vector. For example, a vector of dimension 200. Similarly for sentence embedding, concept embedding, etc. So to learn the semantics, it has many different ways. For example, for word embedding, a typical way to try to learn from its neighboring word. So what does the context of the word to represent the semantics? Why we need to use embeddings? First, it captures semantics beyond exact matching. For example, cancer and tumor are two different words. They have very different lexicons. However, their semantic meanings are very similar. If we use traditional term matching methods, we couldn't find them. However, if we train the word embedding, their related vectors should be projected to the similar semantic space. Embeddings have been used extensively in biomedics domain. For example, for protein function prediction, for drug-drug interaction, and also specifically for biomedical text mining, it has been used for literature discovery. So first, let me introduce Bioconcept work. As I mentioned, it's a concept embedding trained in the biomedical domain. So why we need concept embedding? This is because in the biomedical literature, there are massive amount of concepts mentioned. For example, AP2 is commonly mentioned in the biomedical literature, but it's ambiguous. For example, it can be referred as a gene, it can be referred as a chemical, or even cell line. Similarly, many genes have synonyms. Some genes have more than 10 synonyms. So in such cases, we should have a concept embedding to capture their semantic meanings effectively. How do we do that? So first, we use the full PubMed abstracts as the training corpus. Then we used PubData to recognize 5 entities mentioned in PubMed abstracts. For example, mentioned genes, mutations, cell lines. Then we trained those embeddings in 4 ways. For example, continuous better word or skipgram, that's for word-to-vec. We also trained GLOVE, we also trained fast text. After that, we evaluated those concept embeddings in 9 datasets. It includes intrinsic tasks. That means we don't train the embeddings, it directly use the vectors built. We also evaluate in the extrinsic. That means it's for the downstream tasks. So together, these 9 datasets have 25 million instances for the evaluation purpose. Due to the limited time, I can't go through the task one by one. So here I briefly show 6 tasks or 6 datasets. So this part is for direct comparison. That means we use the exact same parameters from the existing work. We try to compare them directly, use the same embedding type, same hyperparameters. We directly compare them for fair comparison. Here, the second part is we try different hyperparameters. We try to hyperparameter tuning, further improve the performance. So Basically, so the results on 9 datasets show that our Bioconcept vet has better performance than existing embeddings. We also evaluate them into 2 downstream datasets. As I mentioned, first one is protein-protein interaction. It achieved an AUC of 0.95, better than the existing methods. We also tried to evaluate concept embeddings in drug-drug interaction extraction. It achieved an F1 score over 0.8, also better than existing methods. So, second, let me quickly introduce our publicly available tools. So, Bioconcept Vet, as I mentioned, is a concept embedding for 5 types of entities mentioned in the biomedical literature trained on PubMed. It's publicly available. BioWordVec, that's word embeddings. We use fast text trained on PubMed and Mesh. BioSentVec, that's sentence embeddings we trained on PubMed and Mimic 3. And we also have a public sentence retrieval server called Leaksets. So you type a query, for example, you type a sentence or passage in the query, and then it retrieves the similar passages or sentences from PubMed and PMC. So all these tools are publicly available. We hope they are helpful. Thank you so much for your attention. Look forward to your comments and questions. Thank you.